This is a demonstration of how to use Reszon DOA rapid drug cup test. Humidity and temperature can affect results. Do not use the test after the expiration date indicated on the package. Do not use the test if the pouch is damaged. The complete test kit contains product insert, Reszon DOA rapid cup test device sealed in foil pouch and key. Check the expiration date before use. Materials required but not provided: clock or timer. Sample collection and preparation. Fresh urine specimen. No special handling or pre-treatment is required. Urine can be collected at any time of the day. Bring the drug cup test to room temperature, 15 to 28 degrees Celsius. Open the pouch only when it is ready to be used. Open the foil pouch at the notch. Label the cup accordingly. Remove cap and fill in urine to above the minimum level. Screw the cap properly. Verify that the urine temperature falls within the normal range, 32 to 38 degrees Celsius or 90 to 100 degrees Fahrenheit. Using the key provided, turn the lock clockwise about 180 degrees with key until it tightens. Place the cup on flat surface. The urine specimen will flow to the lower level of the cup and then into the test device well. Set the timer to 5 minutes. Read the result within 5 minutes. Important: the result must be interpreted within 5 minutes. Waiting more than 5 minutes may cause the reading to be inaccurate. To avoid confusion, discard the test device after interpreting the result. Negative result. 2 horizontal purplish-pink color bands appear, one in the control zone and one in the test zone. The color intensity of the control line and test line may not be equal. Any faint band appears at the test zone, visible within 5 minutes, should be interpreted as presence of the test line. This is a negative result and indicates that the drug concentration is below the detection sensitivity. Positive result. A purplish-pink color band appears in the control zone but not in the test zone. This is a positive result and indicates the drug level is at or above the detection sensitivity. It is recommended that all positive results to be confirmed by using an appropriate analytical technique. Invalid result. If no bands appear, or a test band appears without a control band, disregard the results. The presence of a control line is necessary to validate test performance. If this occurs, the assay should be repeated with a new test device. Urine specimens may be infectious. Properly handle and dispose of all used reaction devices into a biohazard container. Warnings and precautions. For professional in vitro diagnostic use only. This product insert must be strictly adhered in order to produce accurate test results. Deviations from the procedure may lead to aberrant results. The test device should remain in the sealed pouch until use. Do not use test device if the sealed pouch is visibly damaged. Do not use kit beyond the expiry date. Urine specimens may be infectious. Properly handle and dispose of all used reaction devices into a biohazard container. Wipe any spills of specimen promptly with disinfectant. Do not reuse test device. Limitation of the test. This product is designed for use with human urine only. Although the test is accurate, there is possibility that false results occur due to the presence of interfering substances in the urine sample. The test is a qualitative screening assay and is not for determining quantitative concentration levels or the level of intoxication. Adulterants such as bleach or other strong oxidizing agents, when added to urine specimens, may produce erroneous test results regardless of the analysis method used. If adulteration is suspected, obtain another urine specimen and retest.